In this video, we are going to review passive transport and talk in more depth about the three types of passive transport: simple diffusion, facilitated diffusion, and osmosis, the diffusion of water. So what I want to start with is a quick review of passive transport. So remember, molecules are going to move from areas of high concentration to low concentration. That means they're going to go from where there's a lot to where there's very few. And I think of this as like how introverts like to operate. They're going to go from where there's a lot of people to where there's fewer. Okay. And so this is going to happen passively. It's not going to require an input of energy or ATP. So let's start with a talk on simple diffusion. Okay. I want to take a look at this first image, and it's a time lapse. So we're going to start here on the left side. Notice there's a lot of this particular molecule. Okay, there's a lot of it outside the cell, and there's very little or none in this case inside the cell. So that's the concentration gradient. There's differing amounts on either side of the plasma membrane. With passive transport, molecules want to go from where there's a lot to a little. So you'll notice that over time, two of them have moved to where there's less. And then eventually it's going to kind of equal out to where molecules have moved from an area of high concentration to lower concentration. So you'll notice that no energy was involved and there were no transport proteins. These molecules did not need any help crossing this plasma membrane. And that's because molecules that can use simple diffusion tend to be small hydrophobic molecules. Okay. And because they're hydrophobic, they're able to cross this hydrophobic barrier. Think of like goes with like. Hydrophobic molecules can cross a hydrophobic barrier. Okay. Facilitated diffusion is the same in that molecules are going to go from high amounts to low amounts, and there's not going to be an input of energy. The difference is this idea of facilitated. What does it mean to facilitate something? To help, right? And so that means in order for this to happen, there's going to be this helper protein. It's called a transport protein. Notice it's a transmembrane protein spanning the entire plasma membrane. And notice that it's hollow in the middle. This is going to be what allows molecules to go from one side of the membrane to the other. So let's look at the green molecules first. If you count them, you'll notice that there's more on this side of the membrane outside of the cell, and there's fewer inside. So the molecules are going to go from high amounts down through the transport protein to where there's less. And again, no input of energy required. The red molecule is doing the same, right? It's going from areas of a lot to areas of a little, and it's going to be able to cross through the transport protein to the other side. So these molecules need help because they tend to be small hydrophilic molecules. Remember, hydrophilic means water loving. So they are not compatible with the hydrophobic barrier that this lipid bilayer creates. Okay, so because they're not compatible, they need the help of this transport protein to get across. The last type is going to be osmosis, which is the diffusion of water. And there's a lot happening here. So let's look at just the far left picture. Okay. And so here we have a term, hypertonic solution. And so what we're doing is we're looking at the solution to see how much of a solute is present. So remember, solutes dissolve in water. So we're thinking of things like salts or sugars. In a hypertonic solution, there's going to be more solute in the solution compared to inside the cell. Okay, so this circle is the cell. Think of this maroon colored boundary. That's going to be the plasma membrane. And so in a hypertonic solution, focus on the word hyper. If you say, man, I'm feeling hyper right now, that means you have more energy than usual. So a hypertonic solution is going to have more solute compared to the cell. Now, when we're thinking about water, water is the universal solvent. It loves to work. It loves to break things down. Okay. So water is going to go where there's solute to dissolve. So in this case, there's more work or things to solve outside of the cell. So if a bacterial cell is placed inside of a hypertonic solution, that water is going to rush out of the cell because it wants to start dissolving this solute. So what's going to happen to the cell if water is rushing out? The cell is going to kind of shrivel, okay? We use the term plasmolysis, and that's because bacterial cells have a cell wall. Okay, this cell wall is very rigid, very strong. It maintains its overall shape. So, where the quote unquote shriveling comes in is on the inside. Parts of the plasma membrane tend to detach from that cell wall. So, the inner surface area of the cell is shrinking because water is rushing out of that cell. Okay, so the term is plasmolysis. The overall cell itself does not shrink. The cell does not get any smaller. Just the internal surface area does as that water is rushing out. Okay, now let's look at the far right, which is kind of the opposite scenario. This is called a hypotonic solution. And with a hypotonic solution, there's less solute, like salt, outside of the cell compared to inside. Okay, so we see this concentration gradient here. But we're focusing on water, okay? Water is always going to want to go where there's more work to dissolve. And so when a bacterial cell is placed inside of a hypotonic solution, water is going to rush into the cell because there's more stuff to dissolve. Okay. And so in this case, the cell is going to swell. Okay. Now, it takes a lot of water to rush in for us to be able to visibly see it because, again, that cell wall is very rigid and it helps the cell maintain its overall shape. But with enough water rushing in, it can swell. But notice we're not saying that it's going to lyse. Can it technically lyse? In certain conditions, yes, but generally that's not going to be what happens. Okay? So what if we place a bacterial cell in a solution that has equal amounts of solute inside and outside the cell? We call this an isotonic solution. In this case, water is just going to naturally enter and exit the cell at the same rate. There's no rushing of water. Okay. Now, what you might be asking me is you're like, Chelsea, water is going to where there's lots of things, right? But I thought with passive transport, molecules move from high concentration to low concentration. This is where the definition of osmosis comes into play. And it is where water goes from high amounts of free water to low amounts of free water. And you might be like, what's free water? That basically means that there's water that's free to do work. Okay. And so for me, just the easiest way to think about it is just that water is going to go where there's work to do. Okay. And so there's more molecules, like free molecules of water. If I'm looking at this hypertonic side, okay, the left-hand picture, do we agree there's more free water inside of this cell? Because there's not much work to be done. There's not a lot of things to be dissolving. So I can kind of picture water like twiddling its thumbs, being like, "Ooh, I wish I had some stuff to dissolve," right? There's lots of free water over here. Compared to outside the cell, there's less free water because there's more work to be doing. More water is busy trying to dissolve this solute. Okay. So in this scenario, because it's passive transport, there is water moving from where there's high amounts of free water to low amounts of free water. Again, because there's less free water outside of the cell because there's more things for it to be dissolving. Okay. All right, y'all, that wraps up our conversation on passive transport. As always, let me know if you have any questions.